Early cancer cells are difficult to detect by the immune system because they still resemble normal cells, and they have not developed large masses. Also, tumor cells are fairly non-inflammatory. However, as the tumor cell grows, the immune system can begin to detect it and get rid of the foreign body. Pretty much all immune cells are equipped with fighting cancerous cells in some way. Immune cells can eliminate cancers once it identifies it as non-self using MHC or other similar receptors. However, cancer cells can learn to mutate to avoid detection by the immune system.
In the year 2020, Dr. Mike Effern from the Institute of Experimental Oncology at the University Hospital Boone discovered that one way cells avoid the immune system is by changing their appearance and releasing molecules that suppress the immune system.
Major histocompatibility complex is a receptor expressed on human cells to help the immune system determine if a cell is self or non-self. MIC is a class that is expressed on cells when they're stressed out, such as when they are cancerous. Natural killer cells recognize the MIC on tumor cells using the NKG2D receptor, but often cancer cells cleave the MIC receptor to avoid detection. They also express the NKG2D ligand to activate natural killer cells, but they often cleave that, and the NKG2DL released is immunosuppressive. The immune responses Th1 and Th2 are involved in tumors, whereas the Th1 response is tumor suppressing and the Th2 response is tumor promoting. The Th1 response fights intracellular pathogens like viruses and also cancer cells, while the Th2 response fights extracellular pathogens like bacteria and parasites. Th1 cells activate CTLs and macrophages through interferon gamma. And Th2 cells activate B cells and antibodies through interleukin 4. Tumor cells during the Th1 response are targeted by CTLs and macrophages and are killed. The B cells and antibodies in the Th2 response are not going to kill the cancer cells. Immune responses are regulated by positive feedback of the secreted cytokinins.
As talked about in class, the Fas ligand, or Fas-L, is a molecule that binds to a Fas receptor, normally inducing apoptosis of a cancer cell through a series of reactions. New studies have shown that in some cancer cells can learn to express the Fas ligand. This can induce the Fas pathway against immune cells, which kills the anti-cancer cells in a counterattack fashion. This can cause a buildup of immune cells to the area, but the cancer cells still won't die.
We will now be talking about possible treatments for immune system evasion. Tumors can display very specific antigens on its surroundings, and these antigens can be isolated in a lab setting. Antigens are very specific to these tumors. A cytotoxic drug can then be developed, which would be a drug that kills cells, and be attached to the antigen via a link. These modified tumor-specific antigens can then be put back into the organism and deliver the drug effectively only to tumor cells, thus causing the drug to kill the tumor.
In conclusion, the immune system can detect healthy cells by receptors on their surface. Cancer cells can mutate these receptors to avoid being seen. Cancer cells can cleave MIC to avoid being detected. In addition, they can cleave their NKG2D ligand to release immunosuppressive molecules. Th1 responds to intracellular pathogens, which can kill cancer. Th2 responds to extracellular. Cancer cells will trigger activation of Th2 to be able to survive. Cancer cells can use the Fas ligand themselves to kill immune cells.